Good morning everyone. Today I'm going to give presentation about Salmonella bacteria, and I'm going to cover topics like structure of Salmonella virulence, factors in human host, mechanism and immunological response, laboratory diagnosis, critical analysis about genetically diverse and community acquired infection. Okay, let's go to the topic. What is Salmonella? Salmonella is a type of bacteria that can cause diarrhea illness in humans. Sometimes it can cause abdominal cramps and vomiting too. This is infection have mild symptoms and recover without treatment, but some people require care in hospital setting. Structure of Salmonella bacteria. Salmonella belongs to the genes of rod-shaped gram-negative bacteria. Salmonella is a rod-shaped bacteria. Also, is a gram-negative bacteria. The family belongs to Enterobacteriaceae, and the class is Gamma Protobacteria. There are three type of species. The first one is S. typhi, and the second one is Enteritidis. The third one is Chlorosis. Three species. Here you can see the three species and the reservoirs of the host preference. The first one Chlorosis, the preference for animal, and the second one S. typhi for human. The third one for humans. And Salmonella are gram-negative flagellated facultative anaerobic bacteria that process three main antigen. H is the flagella antigen, O is the somatic antigen, and VI antigen. H antigen has two forms, which is phase 1 and 2. It can change from one phase to the other phase. O antigen occurs on the surface of the outer membranes and can be determined by some specific sequence and the surface of the cell. VI antigen is a superficial antigen overlying the antigen O, and it is present in a few of the serover, especially in S type. Structure of Salmonella. Let's have a look at the structure of Salmonella a little bit more deeper. Antigenic structure of Salmonella. Somatic O or cell wall. These antigens are highly alcohol resistant and heat stable. And VI antigen present in only 3 serovaires of Salmonella. These antigens are heat liable antigens. Now we can have a look at the basic structure, which is LPS. LPS consists of three parts: an outer layer of O polysaccharide coat and middle portion and inner lipid. LPS. The cell envelope consists LPS. The LPS is made up three components followed by an outer O polysaccharide coat and the middle part and the inner lipid A part. O polysaccharide chains of the LPS can vary both in structure and function, providing biological identity to each of the serovaires of Salmonella enterica. The R core is most important part against which the antibodies can directed. Inner lipid A coat help responsible for the toxicity of gram-negative bacteria, also called endotoxin molecule. Here you can see the lipid A and the middle core portion, and the first one is outer layer, and the first one is flagellum H antigen and the VI antigen, and here O antigen. This is the cytoplasm. Now we can have a look at the virulence factor. What are the virulence factors? Certain factors are produced by the microorganism that helps in evoking disease. The virulence factors like adhesion, inversion, and the other toxic genes, some cluster in some of the areas of the chromosome and an old large plasmid. A number of virulence phenotypes in Salmonella have been mapped to the pathogenicity island. 21 SPI, Salmonella pathogenicity island, has been found, but SPI 1 and SPI 2 plays a role for the inversion of the non-phagocyte cell for the multiplication. and the replication in the phagocyte as well as non-phagocytic cells. Salmonella pathogenicity island. This is the figure of the Salmonella pathogenicity island. Here it is lysosome and oxidative stress and antimicrobial peptides and apoptosis and inflammation induced by macrophage. The SPI1 bacterial transportation into the cytosol. SPI1 is a 40 kb DNA region which encodes type 2 secretion system. It helps in transporting the bacterial protein into the cytosol of the host cell. This causes a cytoskeletal rearrangement which helps in the uptake salmonella via a membrane-bound vessel. It mainly helps invading the epithelial cells of the intestine. And SPI2 PI2 encode TTS injects the effector protein for the biogenesis of SCV inside the cell. SPI2 is also 40 kb DNA region that is encoding a second type 3 secretion system. This gene is required for the intracellular maintenance of the bacteria. Inside a membrane compartment, components of SPI2 affects the sorting of the membranes of the phagosome as well as the recruitment of the oxidose to the membrane of the phagosome. SPI3. SPI3 contains a gene MGTC that is required for the invention in the macrophage. The virulence of the Salmonella encodes 5 different CYPs such as SIP, A, B, C, D and E that are capable of causing apoptosis in the macrophage. Mechanism of antimicrobial resistance in Salmonella species. Mobile genetic elements. Mobile genetic elements like plasmids, transpons and recently explored integrants play an important role in the evaluation and the dissemination of the multi-drug resistant gene in gram-negative bacteria. Class 1, the genomic island, was suggested to be an important aspect for the development of the antibiotic resistance in the bacteria. Class 1 integrants contains an array of one of more resistant gene cascade. A3 conserved segment contains QACE1 that provides partial resistance to some antiseptic, the SAL1 gene that encodes sulfonamide resistant. And class 2, certain genes present in the genetic island undergo mutation to develop antibiotic resistance. Class 2 integrants are related to TNT transport family and has till now been identified in the serovirus, typhimurium and paratyphi B. Mechanism of resistance to antibiotics. Mutation in GYRA gene fluoroquinolones. Fluoroquinolones resistance is due to the acquisition of the mutation in the genes that are normally targeted by the fluoroquinolones. The two main enzymes that is target of the quinolones are DNA GYRASE and topoisomerase of the bacteria. The resistance of the aminoglycoside occurs by the enzymatic modification of the 16S RNA subunit that prevents the aminoglycosides from binding to the ribosomal target. And the second one is gene tetracycline resistant. Resistant to the tetracycline was conferred by the TET-A gene, the gene located in the X-bell fragments. The third one is streptomycin resistant against beta-lactamases. Resistance to the streptomycin has been found to be conferred by DFRA14 gene present in the streptomycin resistant gene. And the last one is PSE1 gene resistant against ampicillin and amoxicillin antibiotics. PSE1 gene secreting PSE1 enzyme gives resistance to the B lactomoses like amoxicillin and ampicillin. And let me explain about the picture. This is aminoglycoside influx and this is aminoglycoside influx. This is the efflux pump, which come from the MITs, and normal ribosome inhibition, and this is ribosome mutation and modification. The host bacteria interaction. The epithelial cells and the phagocytic cells like neutrophils and the macrophages identify the specific pathogen associated molecular pattern as well as the endogenous danger associated molecular pattern molecules present in the bacteria. This triggers an immune response. The flagella bacterial DNA and the LPS activate the signaling of the TLR4, TLR5 and TLR9. These are necessary for the inflammatory response inside the host. During the initial stage of an inflammatory response, the macrophages and the neutrophils are recruited to site of infection neutrophils. Macrophages and the monocytes kills the bacteria by phagocyte. The surface antigen present in the bacteria induce the downstream immune response, recruiting the other cell types. So this is the host bacterial interaction. Immunological response during infection in human host. This is the picture. The first one is flagella. This is the bacteria body, and this is invasive Salmonella serover, and this is epithelial cells. And here this is macrophage and cytokine, and this is apoptosis or macrophage. The membrane structure of the Salmonella helps it to reach the epithelial cells. These membranes help reach to the epithelial cells. Salmonella species crosses the epithelial barrier by the passive transport caused by the dendritic cells. As the bacteria reach the layer intestine, the bacteria adhere to the mucosal membranes and invades the epithelial cells. This occurs in microfold cells of the Peyer's pages. Here you can see the Peyer's pages. And bacteria are engulfed by the proximal macrophages. It secretes the effector proteins into the cytosol. Here is the macrophages, here is the secretion is happening. And Salmonella will proliferate, causing secretion of the cytokine by macrophages. Apoptosis of the macrophages will cause the Salmonella to invade the epithelial cells or the other phagocyte cells of the innate immune response. Here is the apoptosis. Adaptive immune response. Adaptive immune response, also referred as the acute immune system. This is the subsystem of the immune system that is composed and specialized for eliminates pathogens by preventing their growth. The antigen presenting cell produce cytokines. The antigen presenting cells produce cytokines or the protein which are secreted from the cells. TNF alpha, IFNA gamma, IL-1, IL-2, IL-6 and IL-8 are the central mediators. The Kupffer cells. The Kupffer cells is produced in the liver, which cause activation of the CD4 plus cells. CD4 plus cells secrete cytokines that activates the phagocytes. The clearance of the bacteria. The clearance of the bacteria from the tissue is brought about by the CD28 dependent activation of CD4 plus cells. Interaction between B and T cells is required for the activation of the antibody response and ISO type switching of the antibody response against the isotopes. Now we are going to have a look at the laboratory methods to diagnose Salmonella infection. There are 5 methods. The first one is bacterial culture method, and the second one is serological assays and antibody detection viral test and rapid serological tests and molecular assay. Bacterial culture done in a number of selective media like SS agar, bismuth and sulfur agar, brilliant green agar and Xsolus and lysine dixochlorate agar. And the serological assay, an enzyme immune assay for the detection and measurement of serum IgM and IgG and IgA antibodies to Salmonella was developed with commercially available lipopolysaccharide of Salmonella typhimurium and S. enteritis combined as antigen. And the antibody detection viral test. The viral test help in measuring the agglutination antibodies against the flagellar antigen and LPS, both acute and convalescent. Phase sample are taken 10 days apart. A positive test should determine a 4-fold increase in the antibody titer. Continued by the diagnosis laboratory finding test, and here we are going to see the rapid serological test. Rapid serological test, the venous whole blood serum, capillary samples and used for the detection of antibody against Salmonella, the antibody class detection and IgM. Molecular assay PCR, real-time PCR, the another test. For example, type 4 detects IgM and IgG antibodies that are directed against the outer membranes protein of Salmonella typhi. T-Ubex-TF detects the antibodies against the LPS of the Salmonella by an inhibition assay format. For the PCR-based assay, HT flagellin gene, VI capsular gene and the 16S RNA gene are used to diagnose. Factors responsible for the development of genetically diverse Salmonella and risk of community acquired infection. Behavioral factors. Inappropriate prescribing and dispensing of the antibiotics. It is a common practice nowadays in many countries. Doctors, they are prescribing antibiotic without even doing the diagnosis, especially countries like India and developing countries. And if you look at the developed countries like New Zealand and US and Australia, they have a proper protocol and they are following the protocol and they are prescribing the medication. And the second one is attitudinal conduct of the patient. Patient attitudes, belief and expectation about various aspects of their plight, their own coping capacity and the healthcare system influence the reports of pain, activities and disability and response to the treatment. Hospital and the laboratories. Poor hand hygiene practice such as handling a raw animal product with inadequate heat treatment, early discharge of the hospital by chance, changes in practice of lack of ability of the patient to pay contribute to the spreading of the MDR organism, multi-drug resistant organism, and non-human sales and usage of the antimicrobials. Poor regulation of the promotion and the advertisement of the drug facilitates the indiscriminate use of the antibiotics. Now, we are going to look at the genetic factors. There are 3 genetic factors. The first one is plasmid and integrons and transposons. Plasmid, they are self-replicating and extra chromosomal DNA that contains genes for virulence and resistance. And integrons, mobile genetic elements that contains 2 conserved segments that is capable of flanking through a central region where the resistant genes are inserted. The third one is transposon. They are mobile genetic elements which contains insertion sequence and can transpose from one site to the other within the same or different chromosome. Effects of resistance in Salmonella species to public health. Failure in therapy due to limited choice of antibiotics for the MDR Salmonella species. There are very limited antibiotics for MDR Salmonella. Once it becomes resistant, it is difficult to treat with another antibiotic. Increased virulence due to the drug-by-drug combination, posing selective pressure on the microorganism. Drug combination could increase the virulence of the microorganism. It will trigger the posing selective pressure on the microorganism. Increased cost of treatment. These kind of infection increase the cost of treatment in hospital sector due to the high price of sterile product and the injection as well as the treatment. longest hospital stay. This will lead the patient to stay in the hospital too long. And finally, increase multi-drug resistant strains.